Our first speaker is Dr. Balzarotti from the Research Institute of Molecular Pathology in Vienna. He is a leading figure in super-resolution microscopy, best known for his work on MINFLUX imaging technology. Today, he will give a talk titled Accessing the Nanoscale with MINFLUX. Let's welcome.
Thank you. I have already like thanks. Okay, thanks. I think it's that one. Or not? Was it me? Okay. I think we're fine. All right. So I think we should turn that one off. There. I think it's. Maybe if I stay on this side, it works. Yes, I can do it. It's okay. All right. So today I'm going to be telling you about the development of MINFLUX, which is a light-based technique to localize molecules, fluorescent molecules, and I think it has a role to play in the future of structural biology and dynamics. So I think, so from my interaction with biologists, I see that there's a wish for recognizing in situ stoichiometry of protein complexes, conformation of large molecules as well, their dynamics in vitro and potentially as well in vivo. And that's very nice, but there are a lot of challenges to get to that. Basically, you need to have targeted labeling. You need to put fluorescent molecules in the right place. You need high localization precision. You need isotropy because most of the problems are three-dimensional. You need high temporal resolution. By this, I mean in the millisecond, potentially sub-millisecond. It's very hard to, with light, to get further than that because basically you can push things to get a photon every 10 microseconds or something like this, but not more. So, and then, of course, having multiplexing or multicolor capabilities. This will scale up the number of objects that can be observed. And as a general, situation of light microscopy, the human eye can see until the hundreds of microns. The light microscope has this limit in resolution associated to the diffraction of light, and this was described in the mid-19th century. Since the 70s, we have the electron microscope at a very powerful, with very powerful capabilities, reaching the sub-nanometer very comfortably. But still, we keep using light microscopy because its specificity, its high contrast, and its compatibility with live observation. And though in the mid-20th century there was a proposal of a far-field window into the nanoscale through FRET, which is first-order resonance energy transfer, you don't really make images with this. You can just access distances. Then in the mid-90s, the true far-field super-resolution was proposed and gave rise to the Nobel Prize in Chemistry in 2014, 20 years later. And when I joined a postdoc in 2010, 2011 to the Stefan Hell lab, one of the goals was, okay, how far can we take this? Of course, this is a metaphorical arrow. It's not necessarily, I'm sorry, I'm trying to get a hold of there. This is a metaphorical arrow. It's not that you want to get to the picometer or whatever, but this is about how much information you can extract from light-based probing. So the diffraction barrier is associated to how light behaves when you focus it. Basically, if you have a plane wave incident on a sample and you plot the electric field on the surface, when you have a tilted incidence, you're going to see some structure, and the higher the angle, the higher the structure you can see. And if you combine many angles together, you can have very tight spots generated, and these are the tightly focused high numerical aperture spots that you use in a confocal microscope. Now, for visible light, this means sizes of around hundreds of nanometers, 1 or 200 nanometers laterally, and 400 or 500 nanometers in the axial direction. what this means that you excite molecules and you will receive light, but you will not know from which one of them that your photons are coming. And this puts a limitation on the resolution. Still, the super resolution field in cell biology has been touching many corners. For example, in cells, in the nucleus, looking at the structure of chromatin, looking at nuclear pores and transport, looking at many organelles, mitochondria, the cytoskeleton, of course, as well as in bacteria and yeast. So this has been expanding, and this is an example of what these images can, the improvements that these images can produce. For example, from wide field microscopy to super resolved, or from confocal microscopy to stealth microscopy. So there's an improvement that you can appreciate. Now, if you can classify many of the techniques that are around to improve resolution as, recipes to improve the confocal microscope or recipes to improve the wide field microscope, and you can classify them according to their spatial resolution, time resolution, toxicity, whatever. There are many things that you may care about in your experiment. And today I'm going to be mentioning these two. I'm going to be mentioning the single molecule localization microscopy and MINFLUX, which is a localization strategy. A note on single molecule localization microscopy. So to make images, what you do is you get some label on your sample, and you produce stochastic blinking, such that you can have sparse emission of light from molecules that are very far away from each other. You take a movie, and then you localize each one of these molecules, and finally render a single high-resolution image out of the stack of frames that you have acquired. This became very popular because you can implement it in a wide field microscope, and depending on the phenomena that you use for inducing the blinking, this receives many names, but at the end of the day, it's all the same principle. You see on your camera a few pixels, and you feed a model to this image to identify the parameter associated to the center of emission of the molecule. But this has been done before, even before doing imaging of blinking molecules. It was being done for tracking molecules at very high spatial precisions. This is a molecular motor working at the nanometer level precisions in 2003. Now, these two methods have something in common, which is that as they're based on a wide field, see if we get the next. If we get, they're based on a wide field localization, which means that the precision is proportional to the wavelength of emission of light, the numerical aperture of the collecting lens. These two parameters, in a way, define the size of this Gaussian little blob. And then the number of photons is appearing here because the more photons you get, the better fit you can have of a model to this data. And therefore, the precision of the parameters of the position molecule can be of lower variance or lower standard deviation. MINFLUX, the way this way to localize is a bit different and modifies this dependence. Instead of having the wavelength of light, you have an arbitrary parameter, which is geometric, and you can modify. And you can even play some tricks to have an arbitrary exponent here on the number of photons. And I will be talking about that. And the paper that was awarded on this conference was about that. So I want to start with a little physics problem here, which is how to localize a molecule when you illuminate it with a parabolic beam. How do you solve this? So imagine you have a molecule here, you illuminate it with this parabolic beam. So you're going to collect some photon number whose mean is that follow a Poissonian distribution, whose mean is proportional to the intensity of the beam. So you're going to have your Poisson distribution. You may have a lot of physical parameters here that concentrate collection efficiency and everything else that models the physics part of this problem. The Poisson distribution may have whatever mean, And then if you want to know where the molecule is, you need to write the likelihood problem, the likelihood function of this problem, which in this case for a Poissonian distribution is just going to be minus the mean plus n log of the mean. Very simple stuff. And that's great. So if you want to localize this, normally you follow the maximum likelihood approach, where you find the position of a molecule that will make your measurement the most likely. To generate an estimator that does this, what you do is you can just take the derivative of this with respect to the parameter that you're looking for and find the maximum. And for this very simple thing, you just get the square root of the number of photons, because that's everything that there is there. Now, this is great. You localize your molecule. So if you measure 0 photons, your molecule is probably here. If you measure more photons, your molecule is very far away. Now, how precise is this estimator? How can you address that? And in statistics, there is a tool that lets you look into that, which is called, or you can put a bound to the precision of this estimation, which is called the Cramer-Rao bound. This it comes from an extension of the Cauchy-Schwarz inequality in a triangle. And basically, you just need to rename what these elements are. For example, if we say that x is the position of the molecule, is an estimation, which is dependent on random numbers of photons that I have measured. X is going to be a random variable as well. That's great. Then, as I have random variables, this is the internal product that is reasonable to choose is the covariance. And already there, the inequality has on one side the norm of x, which is going to be the variance of the estimation that I'm looking for. So that's great already. Then, if you choose y to be something smart, in this case is chosen to be the derivative of the log likelihood function problem, you can work out that this internal product here is 1, is always 1, no matter what you do. And the only thing that is, and of course you need to compute this, it's a bit long, but it's 1, it can be demonstrated. And then what remains here is what you need to actually compute to put a bound to the precision. And this quantity here is down here is called the Fisher information. It's basically, in a nutshell, it is the average curvature of the log likelihood of the problem. It's something very easy to compute, and it's very useful to compute. So for this problem that I just put here, if one computes this average curvature of the log likelihood function, this function, it turns out to be a constant. It turns out to be the curvature of this beam. And very curiously, the bound of the precision for estimating under these conditions is constant. It's not dependent on the number of photons. This means that the precision is always the same. This is very, very curious. And what it means is that, well, if I'm very far away, my precision is going to be something, but it's going to cost me a lot of photons. While if I'm close to this area, I'm going to have the same precision, but for a very, very low, basically towards 0 number of photons. So the conclusion about this is that it can be very cheap, information-wise, cheap to be close to a 0. And at the 0 of intensity, the precision is non-zero. So it has to be larger than something. So these two results, I find them very, very curious. Now, how do you make this happen? Because this is very nice, but in a realistic scenario, you will not know the value of this. So you need something a bit more pragmatic. And that thing a bit more pragmatic is to use a ratiometric approach. So what you do is you excite a molecule with a beam, you move your beam, the animation is running a bit not well, but we'll see. You make a second exposure, you collect another number of photons, and finally, with these two numbers, you should be able to pinpoint where your molecule is. There's an analogy to this that you can make, where you can think that there's a ruler, where the size of the ruler is the distance between a 0 of intensity to the next one, and the number of divisions is the number of photons you collect. So for example, if you happen to collect 4 photons, these are all the possible measurements you can ever make. And if you were standing here, you would first collect 0 and then all the rest of the photons. But if you were here, you would collect the 50-50 case. So every point in space gets labeled with a photon combination that you can measure. So if you happen to use more photons, you're going to have your measurements more densely distributed. And this is a proxy for higher precision. Now, the trick that we can use in MINFLUX as this ruler that is created is purely geometrical, is that you can keep using few photons, but you can shrink the size of the ruler by putting these two sequential beams closer to each other. And in that way, you can keep using few photons, but your density of measurements that are labeling space are still very high and they're close together, and you have your high precision. So if you do real math with this now, each photon emission is going to be Poissonian. It's going to be proportional to the intensity that excites the molecule. The next exposure will be the same, and you can have this function, which is the fraction of photons that you collect in one of these exposures, as proportional to the fraction of intensity. This function that is here is some sort of sensitivity function, and if you plot it as a function of the position, it's a function that goes from 0 to 1 within the range that you have decided, this L. And what this means is that, for example, if you want to actually localize, you may measure 60 photons first, 40 photons after. You measure a ratio of 0.6, and then you go to this P of x and find x, and this is going to be your maximum likelihood estimation. Still, this thing here is going to be a random variable because it's made out of a ratio of these random variables. It's going to be, upon certain conditional probability, it's going to be a binomial distribution. And this binomial distribution is going to be mapped to the real space through this P function. So this function is mapping photon statistics to spatial localization statistics. And this localization distribution is what we're interested in here. We want to know if this is better than other localization strategies. And for that, we can apply the Kammerer bound. So we want to know how narrow this can be. And for that, one can plug in the Poissonian distribution of the count, the geometry of the beams, compute the Fisher information, and if you do that, it turns out that you get this very innocent-looking quadratic function here that if you evaluate near the origin, you get this very nice surprise. The precision around the origin, instead of being dependent on the wavelength and numerical aperture and these things that you have in camera localization or in stellar microscopy, just has this thing there. And it has this thing there because I didn't put the wavelength and numerical aperture in my model because I didn't have to. That's the whole point. So this means that I can improve my precision by just shrinking this value. And in the analogy that I said before, it is like shrinking this ruler, and the function that is mapping photon statistics to spatial statistics becomes sharp. This is the trick. That's why we call this maximally informative luminescence excitation, because we are manipulating the Fisher information of this problem in a geometric way. Interestingly, everything that is in meters is divided by this L parameter, which means that this function is invariant to spatial scaling. So you can shrink the distance between the beams, zoom in, and increase the power, and this will keep working as long as you don't have too much background or your zero is bad. So in a nutshell, that's the reason why the precision can scale geometrically, and this opens a lot of doors. These doors, of course, if you want to do this in 2D, what do? You build a microscope with some excitation, you pattern the excitation, you put a confocal detection as well, and if you want to image a little object like this one, you're still gonna work with one molecule at a time, and on a camera, you would see something not like this, but more like this, or even aberrated, or a random realization. You don't care about this, you just care about how many photons are here accumulated. And what this means is that if you collect how many photons this little molecule here, excited by this parabola, gets from here, from there, and there, and there, you can, again, write the likelihood function for this problem and localize your beam. So this was demonstrated experimentally, very successfully. The precision of localization versus number of photons scales with the square root of the number of photons, but it can be shifted downwards by this parameter. You can have a 5-fold improvement in precision or a 20-fold reduction in the needed photons that you need for a given localization. And all in all, it is tunable. When there is background, you cannot shrink this L as much as you want, because then you have background versus background, so there's not more information. And of course, this is the more you want to get, the smaller the region where this works, and is as anisotropic as well. So the important message here is that this is independent of the wavelength and the numerical aperture. It is not based on a photophysical saturation, and it has a tunable spatial temporal resolution that you can use for many goals. Those goals can be nanoscopy. So if you have linking molecules, this is molecules arranged in a DNA origami. Your data is going to look like this, where each one of these traces corresponds to a donut excitation located at each one of these points. And if you just localize each one of these events and make it through the histogram, you get the object that you designed. This is DNA origami, where molecules are placed very precisely 10 nanometers apart. And you can reach the 1 nanometer localization at high signal-to-vacuum ratios with around 500 photons. This is doable. Then if a molecule moves, you can turn your photon traces into a trajectory. And we've done that with DNA origami as well, oscillating DNA origamis. And this worked well. We pushed the resolutions of around half a millisecond with 150 photons, and very decent. And of course, if a molecule goes out of the good region, you can put a feedback loop to keep the molecule in the center. So your photon traces would look like this, some oscillations, but then the central exposure has a 0 here, would always be lower in intensity because you're keeping a molecule in the center of the pattern. And we've applied this to track the small and large subunits of the ribosomes in E. coli successfully as well. This is basically in equal conditions. With a camera, you would get 10 frames in average for one of these molecules, but with MINFLUX, you get 700 localizations. So this is the trajectory of a single molecule. So it's very rich, and it's so rich that then you can compute the diffusion coefficient as a function of time for each molecule that you are observing. So very powerful. This work in particular, this part was tuned towards time resolution and not towards spatial resolution. We didn't care so much about that. So we were using 10 photons for localizations at a 50 nanometer precision, but at a 8 kilohertz repetition. So this was kind of what we did. So there are many strategies with which you can do MINFLUX. What I show right now is that you can expand this to 3D if you have a beam that has a 0 across the axial direction. And what this means, or a result of this, is that the axial precision and the lateral precision become decoupled from each other, and you can tune them independently. Additionally, something you can do is localize with some structure of beams, get a position, and then change your beams, or change their position, and localize again, again, and again. This is what we call iterative MINFLUX, which can cover larger regions, and by larger I mean just the size of a focal volume, which leads to be able to make images in arbitrarily large regions. The precision of iterative MINFLUX ends up being proportional to a number in meters up here, nanometers, and the number of photons get an exponent. I will tell you why. But the data looks like this. So what you do is you create you make your sample, you label it with whatever strategy, and then you can photoactivate. A molecule will appear somewhere, and then you produce these iterative localizations on the fly very quickly. This takes less than 10 milliseconds or so. And then when you reach the smallest pattern that you care about, you squeeze out the remaining photon. So this is storm MINFLUX of nuclear pores in cells. The median localization is 1 nanometer, so most of them are below, many of them are above. And the Fourier ring correlation also is very good. Of course, this being fluorescence means that you can use fluorescent proteins to label, and what you see here is the equivalent done with photoconvertible proteins. So there's no dyes there. That works well. And the cool thing about that is that you can do this with live cells as well. So these are nuclear pores on live cells. So why does it work? Why does it do this when you do it iteratively? So if you put some scheme of beams, the precision will be bounded by some number here, some geometric number in nanometers, and how many photons you have used. This point, for example. This band represents the variability within the search. Now, if you do that and then start again centered there, you're going to have a new precision with a new number here because you shrunk your pattern, and you're going to have some N2 number that is here. It's the total minus what you used already. And of course, at this square root law here will have an asymptote on n1, so this will go down very quickly. Then you can do this again and again and again, and you can remain, if you're smart about this, you can remain in the non-asymptotic part of this dependence, which leads to have a very, very sharp slope in the precision versus number of photon law. And what this means, for example, is that so the design here is that you need to choose how many photons you're going to use and what else you're going to choose. But let's say that you choose L4 to be equal to sigma 3, right? That's great. So then sigma 3 is what? Is L3 over another number of photons. And L3, you choose it to be proportional to sigma 2, and so on. So when you make this choice, you start multiplying square roots of n one by the other, by the other, by the other. And this is what makes the exponents be very high on the denominator here. So that's very cool, and it's another manifestation of the manipulation of the information. In 3D, as I said before, you can use a strategy like this one, and you can have different samples. In this case, it's that same nuclear pore, which back then led to precisions that were even higher in the actual direction than in the lateral, which was funny. And this is reaching the isotropy with a single objective lens, which is pretty unique for this technique. The precision of localization is very high, and even when you have multiple localizations for a given molecule, you can put them all together, and actually the final localization with all the photons that you are using can easily reach the sub-nanometer always, if you care about it. The thing is that you might not care about getting to the sub-nanometer. To do multicolor, what you can easily do is just use dyes with different Stokes shifts and use different spectral channels here. And therefore, if you add all the photon counts that you get, you can do MINFLUX in the usual way that I showed before, so you can localize each molecule there. But if you look at the spectral counts, and you make a histogram of them, you can see that there are two categories, and you can classify each event to be category green or category magenta, and put color to these images. And this can be done in cells as well, and do two-color imaging in 3D in cells, etc. So the systems are a bit complex. They're not so easy to handle. And these cartoons that we put in the papers are not realistic. The systems look a bit more like this, where the system has many components. You want a decent Wi-Fi microscope. You want to pattern beams and steer them quickly. You want to have single photon counting detection. You want to have stabilization, and of course electronics and the logic that controls all of it. And these systems look a bit like this. There's an entire optical table with a lot of mirrors, and these are all the parts that you need to build one of these with all the perks. Of course, it can be simplified and be smaller, but this is what this is. And for example, the stabilization part, because if I'm pointing to nanometer or sub-nanometer precisions, I need to have a system that is stable to that level. What you see here is our infrared stabilization in Z and in XY. These are images of fiducials. And what you see is their location in space in XY and in Z. And what you saw before was somebody talking, and this is somebody touching the table just like this. So it's really extremely sensitive, but it can be controlled. So all in all, what MINFLUX is a localization strategy in the molecules. It is sequential and it is ratiometric. And at the end of the day, it uses finite points of a PSF. Yes, choosing those points in a smart way. What is not a tracking strategy, because when you do tracking, you put a feedback loop. And MINFLUX is in charge of localizing molecules, but it's not in charge of controlling your system. So anything that is about tracking better or faster can be more about putting intelligence here than here. Here, I think we've done everything that we can. It's not power intensive. It uses regular fluorescence of single molecules. It's not easy to build because you want all these features large, and it's not trivial to process. The necessary elements are to have a spatial gradient of the mean and a spatial gradient of the variance of the measure, meaning the Poissonian photon. That's what's needed, and then you can go. And therefore, the non-necessary elements to play these tricks are for it to be fast. You don't need it to be single nanometer. You don't need it to be single molecule. A few molecules or a larger object. It can be done with scattering because scattering will follow Poissonian statistics as well. It can be any pattern, and it doesn't even need a zero. It just needs this variance. So there's a bit more generality than perhaps you said. And putting this on a timeline, comparing to other precisions and localizations, other strategies with different precisions and localizations. MINFLUX came obviously later based on everything that happened before. It modified the precision in an interesting way. People took this and merged structural illumination microscopy with single molecule localization to get better precisions. And also iterative MINFLUX came in, and then MINSTED, which is basically MINFLUX with the effective TSF of stealth microscopy, is as well there. So, and this is even outdated. There's other things coming up, not from this side necessarily, but achievements on what people are obtaining in terms of performance. So this has been transferred to a company. It became commercial. It's 2019 or 2020, and there are 20 or 30 devices around in many universities and departments. I think there are two or three in China, and also in the US and in Europe. So people are using this mostly in facilities or big centers. And yeah, this has been taking off in the last 5 years. In my group, I'm developing many aspects of this technique on the technology, on the optics that I will tell you about, on tracking more than one molecule simultaneously, which is I think one of the frontiers. There's a lot of software work to be done, and we will be publishing hopefully soon these packages of software that are allowed to, people should not reinvent the wheel. And finally, we're also diving into combinatorial multiplexing strategies to look at flexible molecules like DNA and RNA at this resolution. So also we're exploring this technology because, as I said, sure, I have a molecule, I excite it, and I can use this beam, but I can use many shapes there. What is the optimization process? Where could it take us? And on the detection side, one can have a confocal detector, but one can also have spectral channels, time-correlated single photon counting, and multiple pixels as well. And this makes, instead of having 4 number of photons or few numbers of photons of the main measurement, you end up having hundreds of measurements of photons, which become more sparse, but they may be encoding more information. So a very trivial example of this, but it's still valid because we develop a very large model that accounts for a lot of things, background, all these time beams and so on. We made a simulator that can give camera bounds, can simulate, and can estimate, and it's fed by a vector beam simulator for beams. We actually also closed a loop here from the performance to the vector beam with the PyTorch library, and it works. And push. And this is what feeds the processing package that takes the data from the microscopes and finally renders data. A simple example of this is that if instead of using the same beam, just mix the beam shapes, use these vortex beams and then the top hat beams in the axial direction here, your performance can really improve. So what you see here is that the purple is this one over here and the yellow is this one over here. When you shrink L, you can have double the precision, so half the precision, just by switching the beams. So there's a lot to do with this, and this can lead to very high time. Now, most of the problems that we attack are 3D, and this means that we need to deflect beams in the actual direction. And there's not a lot of practical ways to do that. This is something we attacked. Originally, we used an electro-optical varifocal lens, which is a Kerr crystal, nonlinear crystal that is very temperamental, very hard to use, very expensive, 80,000 euros. also you can break it very, very easily, and time resolutions are not mind-blowing. So, of course, there are deformable mirrors that can be cheaper and kind of similar or slower than this solution. But we came up with a solution that is very, very cheap and has higher time resolution to deflect beams, and this is what we're using in all our systems. So in a nutshell, we're using an interferometric trick. So if you think of a zero of intensity, it's actually a crossing from negative to positive of the electric field. And if you overlap that with a constant electric field, what's going to happen is that your zero is going to disappear. So this is the exact situation that happens in many beams. The top hat beams along the axial direction has exactly this situation. So if you overlap these beams with a Gaussian beam, just directly, just like that, and this is a bit of the real profile, the coherent overlap of these beams is going to look like a bit of a deformed top hat that is going to be shifting this zero back and forth. So the only thing we have to do is to combine these two beams coherently with some control. And the way we did that was with a polarization interferometer. So basically, we just have a beam. We can rotate its polarization very, very fast with an electro-optical modulator, super fast, sub-microsecond. And then you go twice through an SLM. One to make the Gaussian beam, actually you almost do nothing, but you correct aberration. Then a second time to make your top hat beam. So basically, you pattern in one polarization of your beam, one beam of your beam, you pattern one shape, and in the other polarization, you pattern the second shape. Then at the end of this, you just rotate 45 degrees your polarizations and put a PBS, and here's where they interfere. So this is a common path polarization interferometer, very stable. You don't need to maintain it whatsoever, and it can create deflections that look like this that cover around 600, 700 nanometers, which is plenty for doing MINFLUX. What you see here is ESS measures every 200 nanoseconds. Or 400 nanoseconds, so very, very quickly. You see, we can measure the transient really fast. And of course, we can put different shapes there. And most importantly, this double pass configuration has a lot of real estate available in the SLM. So we can have many, many other beams. So what we built actually is a 4-channel version of this, where we have 4 beams that go into the SLM, go out, go in again on second pass after rotating polarization, and come out. And we use this to have 2 different shapes and different colors, which is something we want to use for dual tracking or dual emitter tracking. Additionally from that, there are many other interesting beam combinations that you can produce. So for example, you can have tilted planes of zeros, you can displace them in XY, you can displace them as well, or you can even make these triple zeros. So there's a lot of things you can do, and we have some plans for these shapes. The tracking there. So how does this track? So what you see here is a DNA origami. This is an object made out of DNA that you can use as a probing sample. And what we mounted here is an oscillator on top of the origami. So the oscillator can flip up or down a fluorophore, and that's what we measure here in the Z direction, the axial direction. And this works very, very well. High time resolution, very high spatial resolution. And we can measure transition speeds. Basically, if we push the time resolution really, really, really push it to 50 microseconds, we can still not measure the transition of the DNA. So it's way faster than what we can measure this way. But our system is very, very, very fast. So what's happening in the world of tracking? There's been, of course, historically, single molecule tracking of cameras. There's the first MINFLUX tracking work that I showed before, and the follow-up with origamis. There's been recently, in 2023, 2 works looking at walking motors that became very popular, but all of this is a 1-emitter work. And there's been a 2-emitter, 1-color work where they separate 2 emitters by lifetime, but still in 2D. There's been a 2-color 2D 2-emitter work for walking motors. And what we are doing is 3D two-color two emitters that we want to apply to many, many problems. What these experiments look like is like this. You take a picture of the single molecules. They look like this. They're very bright, actually. And we choose which ones we want to address with a feedback loop, and we track them with a spin, like this one. Once we like where we're positioned, we start interleaving our two excitations and get a trace for the green molecule, a trace for the red molecule in sync. This means 7 exposure for green, 7 exposure for red. This is 14 exposures. And we can just localize. So we can get trajectories XYZ, XYZ for each molecule. This is a test origami where a molecule can oscillate and bind here, here, or here for the red, here, here, or here for the green. We did this on purpose such that they can bind to the same place and make sure we can have, accuracy of our two systems, because this is very hard to do actually. We had to calibrate things to 10 to minus 4 precisions to really reach this ALBA's achievement. And they're quite well overlapping, sub 2. Okay? We can trust it. And we're putting this to use. One of the things we're putting to work is to track the conformational changes of protein complex called cohesin. Cohesin is part of a family of complexes called the structural maintenance of chromosome complexes that has been shown to extrude DNA. So what you see here is this is 2 microns or so. This is large, not even longer. And what you see here is a DNA molecule labeled fluorescently. That's all you see here. DNA. You don't see this molecule, but you see what this molecule is doing, which is it's zipping the DNA. It's presumably walking through the DNA and remaining there as a ring. This is something that shapes chromatin. It's very important to keep the shape of chromatin in many states of cell. And this has been obviously seen by fluorescence microscopy and has been attacked by other techniques. For example, structural cryo-EM. And so in cryo-EM, the issue is that this complex is so flexible that you need to artificially lock it in place to get a stable structure. You can do FRET, but this protein complex is very large besides being flexible. So FRET has a range of 1 to 8 nanometers, but this has 50 nanometers inside. So again, you cannot see everything. And of course, you can do high-speed atomic force microscopy, but it's very, very low throughput, and it has some artifacts of the surface. So MINFLUX is multicolor, multi-emitter MINFLUX is a good candidate to understand what are the steps that this molecule is following to extrude DNA. These are initial experiments, but they look like this. We label 2 sites, so we are very ambitious. We want to label more sites, and we're on that way. So we choose which molecules we want to address, and then we just measure. And what we found was very, very variable movement. So we see that there are very different regimes where the subunits are doing this, then they're going together, then they're rotating together with respect to something else. So this is absolutely, we're starting to process this data, but we need a lot of modeling work. So this is something we're going. And then finally, another flexible object that we're addressing with these techniques, because what I do think is that there's a lot of space for these techniques to work on flexible, inhomogeneous objects that are not addressable with cryo-EM or other. And so what you see here is RNA particles. So if you take a cell and you somehow, it doesn't matter how, you pull the RNA out of it, you may be able to, in an electron microscope, make images of each one of these RNA particles. And what you see here, each one of these is an RNA molecule. And what's painted in green is a protein complex that can be identified by a cryo-electron tomography. And this is very nice, and you can average things, but as you may see, the RNA is averaged to be a sphere because it's so inhomogeneous that you cannot do much. So you should be able, if you want to know what's going on here, you need to have per single object, no averaging, a trajectory or some structural information. So this is the adventure that with 2 PhDs, we're navigating, and the question of can we measure the nanoscale organization of mRNA? And the way we're addressing this is by labeling with a technique called RNA fish, where you create a DNA probe that is complementary to the RNA and will bind it. And then you make this little extension to put a fluorophore there. This is something that is done for 20 years. But what we want to do is a super resolution version. So we want to have blinking of molecules that bind and unbind transiently, such that we can localize each one of these RNA probes. And for that, we can use DNA paint, which is the technique that allows to do that. Here you see a DNA origami image of DNA paint. And the question is, well, which modality should we acquire data? Should we do wide field or should we do, will it be enough? Well, 5 or 10 nanometers are going to be a bit messy for looking at the RNA particles. We're going to need 2 or 1 nanometer to be able to trace or to know its structure. So we do need high resolution. So the way the experiment goes is that you make your primary probe with a stable imager to find your RNA and a transient imager that will do the super resolution. You choose one mRNA and you design all these primary probes that are going to be complementary to different sections of this RNA. You're going to paint the whole thing. And then what you can do is first put your stable imager there to find the RNA molecule in the first place. And then put the other set of imagers that are going to be blinking, coming and going, and hopefully get an image that might look like this. Now, what does that look like exactly? This is an image of a cell under these conditions. We chose one gene, one mRNA specifically. What you see here is this nucleus of the cell, and the cytoplasm of the cell is all around. And each one of these dots is one copy of this mRNA. So this is a wide field image, no super resolution there. If we do super resolution on this, we are going to have something that looks like this. Exactly. So just in principle, the only thing you see is smaller dots. But if we can make rounds of images, and we can look at, we can paint different sections of the mRNA with different probes and start to get a bit of structure. So what you see here is wide field super resolution of mRNA in the nucleus, or proximal to the transcription site, which is a bit more messy, or even in the cytoplasm, where we find it to be more extended. So comparing this to the expectations, first of all, none of these things looks like a sphere. And they're not very small. All these objects are in the hundreds or larger nanometers. So mRNA molecules are flexible and disjoint. They're not super packed. They're not so tight. If you compare this with the state of the art, what people normally do is just image all these red ones, all these red probes, you get a dot. All these blue probes, you get a dot, and all the green ones, you get a dot. So you get 1, 2, 3 positions, and you can measure distance from this to this and this to this. Here, you can get distances from everything to everything. So you can get an idea of the compaction state beyond just a few sites. And of course, we can push this further if we do MINFLUX, DNA paint, RNA imaging, with precisions where now instead of seeing a cloud, you start to see little clusters here, which belong to each one of the primary probes that we have chosen. And if we push this a bit further, of course, as I told you, MINFLUX can do 3D. We can have images that look like this, where we can see, perhaps not so obvious to follow, but what you see here is 2 RNA particles, one here and one here. The one that is here has this magenta, cyan, yellow region, and this is the other one. And we can really get maps of distances, compaction distances across the sequence of the RNA. We can, of course, we're addressing RNAs of different lengths and different number of introns, which are the parts that are spliced right after transcription. So there's a lot coming for this. Now, very important conclusion of this that RNA is compatible with DNA paint, and this was not done before. Wide field images deliver this precision, while MINFLUX can push further, and you can do that to pierce into the structure. And the future, what's coming with this project is to look at splicing, compaction, transport, and export with a bunch of biologists that are working together with us. And of course, putting here as well proteins on these images. This is something we can do as well. And start to paint a more comprehensive picture of the variability of RNA while it is spliced, while it is a mature particle, and of course when it's exported. So there are a lot of changes in this process, and I think this is the right tool to pierce through them. So all in all, zooming back, in my group, our specialty is the tech, but we're also jumping into wet side of things because we developed a lot of combinatorial multiplexing strategies to achieve that I just showed. We're doing, of course, more complex experiments with tracking, as well as many software packages, and the next generation of MINFLUX-like strategy. My institute is the Research Institute of Molecular Pathology. It's based at the Vienna Biocenter, which is a quite large cluster of institutes with 140 groups, thousands of people, a lot of biotech companies around, and a pretty good place to be in, I gotta say. I would like to thank my group that has been doing all this work very passionately and very with a lot of. They're very brave to these projects because they're all very complex and very challenging. And then, of course, I want to thank the committee and the organizers of this conference for choosing this work. Very grateful to be here, and I will be happy to take. Some what theory, sorry? The last. Yeah. Yeah, so the reality is that we touch a bit of everything in my group. I'm interested in the statistics and the math behind that enables all of this. But if you mentioned AlphaFold, you mentioned it. I think that AlphaFold took data from like infinite data from electron microscopy, right? Very orderly data, very well curated data, and made a model that can predict outputs, right? Here, we're not seeing electron densities. We're not seeing residues of amino acids. What we see is fluorophores. So we can always see in this type of techniques what you target. So if you want, throughout the discovery path, this is kind of in the middle. This is not something used for screening because you need to kind of know what you're looking for. I do think that these techniques might be able to play a role, especially in this stuff, to contribute data about kinetics and dynamics that is not available so far. If you want to take AlphaFold and it doesn't give you a movie. It gives you, an initial conformation or a bunch of possible conformations. That's it. But it doesn't give you dynamics. Of course, the ultimate solution there will be molecular dynamics aided by AI or whatever to give you conformations. But still, that will give you everything that this molecule can do. It will not tell you what the molecule is doing. on a cell at a given point. So I think that the role that these techniques can play is guide and filter out from all the possible conformations or structures you can find from AlphaFold or other techniques, well, what is actually happening in a cell at a given moment. I don't know if it's for the recording.
My question is about, I mean, since you're going like higher and higher in resolution, I was wondering, do you see already the effect of the linker molecule and the size of the linker in terms of like the noise? Do you see like how much it wobbles around, and how do you optimize for that?
Yeah. And so the linkage is there, right? And you, first of all, nobody did a single particle averaging analysis of single molecule data to a level where they could extract these variabilities. But these variabilities are there, and I think they are extractable. There's in cryo-EM, there's a technique called cryo-dragon, which what it does is embed. So you have your data set with 100,000, a million particles, and then 2D, and then you make a tomography to reconstruct the structure. Right? In that technique, what you can do is have an intermediate latent space of lower dimensionality than 1,000, and you can identify flexibility and variability in your reconstructed structure. So I think that if one took a structural object like the nuclear pore or something that is meant to be kind of rigid or rigid, and you apply that type of encoder-decoder latent dimension reduction, most likely you're going to be able to see the flexibility of the linker mixed with the flexibility of the object. And the question is, can you distinguish these two? So there are people who work on, look at rotations of molecules and stuff like this, and they do dual labeling to have rigid labeling. So the label is rigid with respect to the thing that you're labeling. I think that if one did those tricks on super-resolution data and compare one data set versus the other with the right type of averaging, you would see a difference. You could distinguish actual flexibility of the complex from remanent flexibility of the link. Now, in many of the kinetic things, we might not care because we're looking for movements that are in the 5 nanometer, 10 nanometer, and the leakage is in the 1, 2. So that will be fine. But it matters. It matters a lot. For this cohesive stuff anyway, we do covalent linking directly. So we don't use antibodies and antibodies. Well, I mean, here, what you localize is who is absorbing the light. This is what you do. So if you get very, very much into the details, you're localizing where the absorption dipole of this chromophore is. That is somewhere inside of the barrel of the GFP. So if you really, really pushed it, you would find that. Now, if this is moving, well, it's going to be an issue. But again, a rigid linkage might be able to show that. And some people have been doing something like this, where they put an antibody and they have many epitopes. So you have a protein with many epitopes. They can see different GFP antibodies. Sorry, different. How was it? No, they have a GFP and they have an antibody against GFP. Then they have a dye on the antibody. And they can see that an antibody is somewhere with respect to GFP and another antibody somewhere. So now, what do you want to do that for? I guess you would want it to correct for the problem. Of course, with the expansion microscopy, post-labeling, you don't have the problem. And that's why I think that the future of all of this is not about imaging, necessarily, though some things are, but it's about the dynamics. This is where it will play the big role.
So we can see the DNA using the current mixed flux system?
Yes, but you need to let me put it like this. Yes, so we're
Yeah.
looking at RNA, not DNA.
RNA.
RNA. But of course, instead of doing RNA fish, you could do DNA fish and look at the structure of DNA. This is doable. A lot of people are doing that at a larger scale at the chromatin level. And that's a business I didn't want to get myself into because it's very expensive, very large, and very crowded.
It's up to 2 nanometer. It's up to 2 nanometer.
That's but that's that's the precision of a probe. Now it's not you're gonna see the DNA 2 nanometers step by step because you need to have your probes there. So still there's some sparsity.
I want to investigate the interaction between ion and DNA.
An ion?
Ion? Yeah.
Okay. Like a magnesium ion?
Yes. For example, calcium ion, calcium interaction between calcium ion and DNA.
So what I can do is that that problem should be addressed with an intercalating dye. Is this double-stranded DNA? Double-stranded DNA or single-stranded?
Single is okay. DNA and protein, polysaccharide.
Because if you have double-stranded DNA, you can use a groove intercalating dye. No,
single.
So single-stranded DNA. Okay, that is tricky. So, of course, you can localize your DNA. You can put a dye on your DNA. You can put a dye on your protein, and you can see them with 2 nanometer distance. Yes, you can. The calcium ion you will not see because it's not fluorescent. So remember, here you need to put a fluorophore sample. And you will only observe the fluorophore. You will not observe the DNA molecule itself. You will not observe the ion itself. You will observe the fluorophores, the fluorescent molecules that you introduce on your system. So to see DNA, you need to put a dye somewhere on the DNA. At the 5', 3', or in the middle. Whatever. You need to put a dye there. And the calcium ion, you will not see it because it's not fluorescent. To be clear, I think that the way you see ions is with electron microscopy, but depends on what you're doing. I don't know if there's. You can tell me exactly the systems that you work with. I can tell you what I know.
Okay, let's thanks Dr. Balzarotti again and take a short tea break. The next talk will begin at 2:15 p.m. Thanks.